Thank you very much, Mara. So my lab, tumour virology, is mostly interested in human papillomaviruses and their association with the development of a number of different human tumours. These viruses are responsible for about 5% of all human malignancies. Cervical cancer is by far and away the most important, and infection with this virus is linked to about 100% of these cases. These viruses are also causative agents of a significant number of other anogenital cancers and a rapidly increasing proportion of head and neck cancers in specific anatomical sites. Overall, papillomaviruses cause around 600,000 new cancer cases each year, and the WHO has recently defined at least 12 different papillomavirus types as being cancer-causing in the cervix. This just shows you a distribution of cervical cancer overlaid on the map of ICGEB member states, and as you can see, it's a particularly devastating problem within the developing parts of the world. Now, the papillomavirus life cycle is intimately linked to the differentiation of the infected epithelium. The virus is believed to gain access to these basal cells through microtraumas in the skin, and then as these cells then divide and differentiate, you see coordinates expression of the different viral gene products, which ultimately gives rise to new infectious virus particles. And this whole process normally takes place over around a 2 to 3 week period. Now, for reasons that are still unclear, certain individuals can show evidence of persistent infection for many years. And it's these individuals, this is the single biggest risk factor for ultimate progression towards the development of malignancy. And a standout feature of these tumours is the continued high-level expression of the two viral oncoproteins, E6 and E7. And it's these two proteins which continue to drive the development and maintenance of these tumours. And indeed, there's a whole variety of studies that have been done over many years that have shown that if you can block the function of these two viral proteins or inhibit their expression, you can actually induce cell growth arrest, apoptosis or senescence in both the tumours and in derived cell lines. So both of these viral proteins represent excellent targets for therapeutic intervention in HPV-induced malignancies. So we all know that there's actually a very good vaccine out there at the moment, which are very effective. So it begs the question, why are we still interested in therapies for papillomavirus? Well, if vaccination was implemented globally today, it would still take 10 to 20 years to impact on the disease. We have major issues in terms of both cost and logistics in the countries where we really want to get these vaccines to. And it's also very important to remember that these vaccines are prophylactic, they're not therapeutic. So many millions of women who are currently infected with papillomavirus will still go on to develop the disease. And currently there's around 500,000 cases per year, and the WHO projects this to increase to around 720,000 over the course of the next 10 years. And the majority of this increase will be within ICGEB member countries. And also it's important to remember, as I mentioned at the beginning, there are many papillomavirus types that are cancer-causing, and the current vaccines do not actually cover most of these types. So my lab's interest is focused particularly on one viral protein, the E6 oncoprotein, and this just shows you it's a highly multifunctional protein. It interacts with many different cellular targets. And not surprisingly, with such a broad array of interacting partners, it displays a wide variety of biological functions. These can be related obviously towards the viral life cycle, such as activities related to genome amplification and maintenance, but also activities that are related to its ability to cause cancer, such as cancer induction in mice and in vitro cell immortalization phenotypes. Now, one of the key features about papillomavirus is there's actually close to 200 different human types have been described, but not all of these cause cancer. In actual fact, only a small subset of so-called high-risk types are actually cancer-causing. Examples of those would be HPV-16 or HPV-18. In contrast, the low-risk papillomavirus types are only associated with benign disease, and classical examples of those would be HPV-6 or HPV-11. So one of our major interests has been to try and understand why certain papillomavirus types cause cancer, whereas other types do not. And when you go and look at the papillomavirus E6 oncoprotein, it actually has a unique characteristic of being able to interact specifically with a subset of cellular targets that are only bound by E6 oncoproteins derived from the cancer-causing papillomavirus types. None of these are seen by the so-called low-risk E6 oncoproteins. Again, this set of interactions is associated with a variety of different biological activities. Again, activities related to life cycles such as genome maintenance, effects on differentiation and genome amplification, but also, most importantly for our point of view, also these associations are important with a point of view of the ability of E6 to contribute towards cancer induction. So what is the molecular basis for making these statements? What I'm showing you here is, a group of different papillomavirus E6 oncoproteins. This is the extreme C terminus of these proteins, and what you can see that there is a very highly conserved stretch of 4 amino acids at the extreme C terminus of these cancer-causing E6 oncoproteins. And this is a so-called PDZ binding motif, and it is this stretch of amino acids that confers association with these high-risk specific targets of these E6 oncoproteins. If we look at the low-risk E6s, which don't cause cancer, they do not have this interaction motif. So this is like a kind of molecular signature of oncogenic potential in the high-risk papillomavirus E6 oncoproteins. We can make specific mutations in this region of E6 and leave all the rest of E6 function intact, and then ask, what does this do with respect to biological activity? Well, we know that in transgenic mice, this region of E6 is critical for its ability to cooperate with the other viral oncoprotein E7 in inducing tumours. We know that in a variety of in vitro tissue culture assay systems, it's also important for the ability of this viral oncoprotein to cause cell transformation. And most importantly, in the viral life cycle, and don't forget that these activities have evolved to favour viral replication initially, and so it's important to see that also it plays a critical role in the capacity of the virus to replicate in a differentiating epithelium. And in actual fact, if you make a single point mutation in this region of E6, in the context of the viral genome, the virus cannot actually maintain itself in the infected cells. So it plays a critical role in a variety of aspects of E6 function. As I mentioned, there's a lot of different targets have been described, but I'd like to just focus our attention on two of them, two proteins called DLG and Scribble, which are key regulators of cell polarity and are both classified as tumor suppressor proteins. Now, cell polarity itself is controlled by this tripartite complex, and we have Scribble and DLG that define the basolateral region of the cell. We have the PAR complex, which defines the subapical, and we have the Crumbs complex, which defines the apical region of the cell. And through a series of mutually antagonistic interactions, and this establishes a concentration gradient of these different proteins, and their correct cellular localization and quantity is critical for maintaining the functioning of the whole. This then connects to a variety of other critical signaling pathways, and the end result is that it means that the control of cell polarity is linked directly to the control of cell proliferation. And the E6 oncoprotein impacts upon this pathway in a variety of different manners. Now, dislodged DLG was the first target to be identified, and E6 is known to associate with this protein through association with a PDZ domain that's embedded within the middle region of disc large. And one of the one of the fascinating features about E6 is that it actually recruits the cellular proteasome machinery to many of its target proteins and directs their degradation. And that's exactly what it does with disc-large. 18E6 very efficiently targets disc-large for degradation. 16E6 is also effective, but not quite as good as 18. If we can make a single point mutation in the 18E6 oncoprotein that kills the association, it also then has no effect on disc-large levels. And likewise, if we take an E6 derived from a benign papillomavirus type, which lacks this so-called PDZ binding motif, this has no effect on disc-large levels. And these effects on protein level are reflected in the strength of association between E6 and this particular target. You can see here that 18E6 combined very, very strongly to DLG, 16E6 is somewhat weaker. And this was really rather surprising. If you look at this C-terminal sequence that is responsible for this association, you can see that HPV18 and HPV16 are actually very, very similar, except that 18 has a C-terminal valine and 16 has a C-terminal leucine. And we asked the very naive question whether or not this single amino acid difference might be so important for substrate selection. So what we did was to swap those last two amino acid residues, so we make HPV16 look like HPV18 and vice versa. And then when you do the assays, where you look at DLG in the presence of 16E6, you're looking at the ability of 16E6 to degrade this protein, you can see it's sort of effective, it does have an effect, but it's never as dramatic as 18E6. If we make 16E6 look like 18 by swapping that last amino acid, it becomes much more effective. Conversely, 18E6, if we swap that last amino acid and make it look like 16, it becomes much weaker. So a single amino acid difference at the extreme C terminus of this E6 oncoprotein is having a profound effect upon the ability of this viral protein to recognize its different cellular substrates. And so this actually drove us then to try and understand in more detail how E6 is recognizing its different substrates. And what we did was enter into a collaboration with Xiaojing Chen, the University of Colorado, where we obtained crystal structures of E6 bound to its different PDZ domain containing substrates. And this told us critical information about key residues within the E6 oncoprotein that were responsible for this substrate specificity. Now, obviously, this also can be used in a variety of other ways. There's other uses that you can make of this information. One of them is to do in silico screens to see if you can identify compounds that can block the E6 PDZ association. This we did in collaboration with Amit Sharma, where we've identified a large number of potential inhibitors of E6 association with this class of targets. And this just shows you that some of these are actually quite potent at inhibiting interaction between E6 and its PDZ domain containing substrates. The other thing you can do with this information is to go back to the target protein itself, because you know precisely the molecular details of how E6 interacts with these proteins. And you can make mutations such that you render the target proteins resistant to E6-induced degradation. And this is what we've done here. You can see this wild-type protein is degraded very effectively by the E6 oncoprotein. The point mutant is no longer bound by the E6 oncoprotein, and this is resistant to E6 degradation. So the question is, what happens biologically? You can take these mutants and the wild-type protein, you can put them back into HeLa cells. These are cells derived from a cervical cancer which harbor papillomavirus E6 sequences. You put the wild-type protein in, it has no effect, the cells still grow. However, if you take the mutant protein, which is resistant to E6-induced degradation, you can see you have a dramatic inhibitory effect upon continued cell proliferation. So targeting these proteins seems to have a major effect upon the ability of E6 to continue to drive the proliferation of tumor-derived cells. It's important to remember, however, that this region of E6 is actually really very multifunctional, like a lot of these viral proteins, and association with PDZ domain proteins is not constitutive. In fact, if you look at the C-terminal sequence, you can see at the -2 position, we have this threonine, which is actually a phosphoacceptor site. And depending on which papillomavirus it comes from, this can be phosphorylated by either protein kinase A or AKT. And this phosphorylation is important. If you look at 18E6, when this has been phosphorylated by pKa, you see there's a dramatic decrease in its ability to interact with this PDZ domain containing substrate. So does this phosphorylation confer any other activities? What does it, other than being a negative inhibition of this substrate recognition? Well, what it does, if we have a phosphate group on this threonine or serine at this -2 position, it actually creates a consensus recognition site for 1433 proteins. These are molecular chaperones which play key roles in the regulation of the subcellular distribution of their different substrate proteins. And then if you look at the E6 oncoprotein, it's quite striking. If we look at the levels of E6 phosphorylation in cells in different phases of the cell cycle, you can see that the E6 oncoprotein is not phosphorylated constitutively. It actually is most active during the G2M phase of the cell cycle. And this is actually reflected in the substrates with which E6 can interact with, where we see that association with these 1433 proteins in this co-immunoprecipitation occurs most strongly during the G2M phase of the cell cycle. In contrast, if we look at the ubiquitin ligase that E6 also associates with, this is the protein it recruits to target substrates for proteasome-mediated degradation. This interaction is constitutive throughout the cell cycle. So just to summarize what I think, what one aspect of E6 function and how it's contributing towards this, this development of malignancy, we know that E6 association with PDZ domain substrates plays a critical role in the expansion of this, this population of cells, which are critical for viral, normal viral life cycle. We also know that as these cells differentiate and the cells enter a kind of G2M-like phase, this E6 protein will then be phosphorylated and be targeting this other substrate of proteins, the 1433 proteins. What we don't know is what is happening when these cells then progress towards the malignant state. We don't know what the status of E6 phosphorylation is in this, and we're particularly keen to investigate this further, since this may well be a prognostic indicator of the potential of the propensity for these lesions to progress towards malignancy. What we do know is that the later stages of malignant progression, once we get towards the invasive phenotype, the ability of E6 to target these different cell polarity regulators plays a critical role in the invasion process. What I'd like to do in the last few minutes is just to go and say a little bit about the other aspect of work that the lab's interested in, and that is what happens at the very initial stages of viral infection. This is when the papillomavirus gains access to these basal cells, and then the subsequent events that occur as these viruses are transported, the genomes are transported into the nucleus of the target cell. Naturally, this means focusing on the virus capsid, and this is the virus itself has two major components. L1 is the major structural protein, and a very characteristic feature about this protein is that it can self-assemble into virus-like particles, and it's this characteristic of L1 that forms the basis for the vaccines that are currently out there. However, the minor capsid component, L2, is the key player for ensuring that the viral DNA makes the safe journey through to the nucleus of the infected cell. And so we've been focusing our attention on the role of L2 in virus infection. So as I say, the virus is believed to gain access to the basement layer through microtraumas in the skin, where it then can bind to its target cell. The virus becomes internalized. We know that it goes through endosomal transport pathways and acidification, where you get separation of the L2 DNA complex away from L1. L1 gets targeted for degradation at the lysosome. The virus then gets transported, the viral DNA then gets transported through a variety of pathways such that it ultimately resides within the nucleus. Now, endosomal transport pathways, this might look very simple, but actually there are a lot of options. There are a lot of routes that the virus could potentially take. And what we've been particularly interested in is trying to understand how does L2 determine this specific route of infection. And so what we did was to do a proteomic screen for L2 interacting partners, and this was done in collaboration with Mike Myers here in the labs in Trieste. And we identified a variety of proteins that could associate with the L2. The most exciting of which was a protein called sortinexin 17. As you can see from this interaction assay here, this binds very strongly to the L2 protein, but doesn't interact with the other major component of the capsid, L1. So this is a specific association, specific interaction between L2 and the sortinexin. So why was this a particularly exciting interaction? Well, sortinexin 17 is a key player in cargo fate determination in early and recycling endosomes. Through this C-terminal region, it can associate with a variety of cargo proteins, and all of these cargoes have a very well-defined interaction motif. If these interactions are abolished, then these proteins all get targeted for lysosomal degradation. Most interestingly, if we look in L2, you can see that it also has this conserved sortinexin interaction motif, and if we mutate this region, then we abolish the ability of L2 to interact with this cargo fate determining protein. The other thing that's particularly exciting about this is that when we look at all the different papillomavirus types, you can see the L2 proteins all have these highly conserved interaction motifs, saying that this interaction is most likely very important for the ability of a wide variety of different papillomaviruses to successfully infect their target cells. We were then interested in trying to understand in more detail these specific constraints of target selection, and we used a variety of inhibitory peptides that we could use to block association between L2 and the sortinexin 17. And we eventually came up with a minimal peptide, which is very effective at inhibiting interaction between L2 and this cargo protein. And obviously, this will be a very valuable tool for trying to understand how this interaction is important, both in the early stages of the viral life cycle and also for papillomavirus transmission. So how does this interaction actually contribute to the infectious process? And what we've done to investigate this is to use pseudovariant technology. And I'm always amazed by this. It's quite incredible. We can express L1 and L2 within 293 cells, together with a reporter construct, in this case luciferase. And you get high level of expression of these two viral proteins and amplification of the reporter. And over a period of time, these will actually self-assemble into so-called virus-like particles or pseudovariants and actually encapsulate this reporter construct. You can purify these particles and then use them in infection assays. And this is what they look like. This is some BPV preps that we made quite recently. And as you can see, these pseudovariants are really very beautiful and show perfect symmetry with respect to compared to a wild-type derived papillomavirus. If you then use these to infect Hackett cells, which is an epithelial cell line, where we've knocked down sortinexin 17 expression, you can see this has a dramatic inhibitory effect upon infection with these papillomaviruses. And likewise, you can make a point mutation within the L2 protein within the sortinexin interaction motif. And again, this essentially greatly inhibits infection with these viruses. What happens to the virus when it cannot associate with this cargo fate determining protein? Well, what happens is that whole virus just gets targeted directly for lysosomal degradation. So this association is critical for ensuring that the L2 DNA complex can escape from the endosome and progress towards the nucleus and escape the lysosomal degradatory pathway. And this is just to show you that schematically, the association with the sorting nexins ensures that the L2 DNA complex progresses in this route rather than progressing towards the lysosome. I'd just like to finish with acknowledging the members of the lab who have been involved in all the aspects of this work. As you can see, we're really very international flavour of the lab. Obviously, excellent collaborations with colleagues in Colorado, Bob Garcia and Xiaojin Chen, also with Amit Sharma in New Delhi, and Martina Begant in Slovenia, who was instrumental in setting up the pseudovariant technology. And of course, not to forget our sources of support. Thank you very much.